I'd like to introduce Silvia. As I said, Silvia found the squid, a little mollusk, in Wisconsin, in the lab of Margaret McFall-Ngai, one of the leading persons in symbiosis research in the world. And Silvia started her PhD work in Margaret's lab in Wisconsin. And then the thesis supervisor decided that Wisconsin is not good enough anymore, and she decided to move to Hawaii. So what a PhD student has to do is, yes, well, either you go on or somehow it's getting complicated. And Silvia went with Margaret to Hawaii, where we met last time, Silvia, I guess about 2 years ago. And in Hawaii, then she finished her thesis in, I think, in summer last year. And then she moved in the lab of Ned Ruby. And he's also analyzing the squid symbiosis system. And there she did a short postdoc before she, over New Year and Christmas, now left beautiful Hawaii and now is in, I guess, snowy Switzerland, and joined the lab of Philipp Engel as a postdoc. Silvia, it's a great pleasure to have you here. You did fantastic work. That's why the reason why I was recommended to invite you during your PhD and your short postdoc with Ned. You have fantastic papers. You had fantastic and novel insight that small RNAs are used, produced by bacteria, are used to influence the host animal. And I think this is what you are talking to us now. The title of your talk is The RNA Signature of Local and Systemic Interactions Between Partners. Silvia, the floor is yours. Please upload your talk. And after the talk, there is the chance to ask questions to Silvia. Please just use the chat room, and then we will forward the questions to Silvia, and you get the answer from that. So please use the chat room if you want to interact with the speakers. Silvia.
Thank you very much, and I'm really happy to be presenting here today. And let me share the screen. Yeah. So today I'm going to be talking about signaling symbiosis, which is something that I focused during the majority of my PhD. And I apologize that I changed slightly the title of my presentation, but I really wanted to reflect what I'm going to be presenting today and focusing on for this presentation. So I'm going to be focusing on particularly on one small RNA from the bacteria called Ssra, and how we've seen that it modulates the host responses during symbiosis. So in order to answer all these kind of questions that we have, we are using as a model the squid vibrio light organ symbiosis, which is a binary system, which means we only have two partners: host, which is here, you can see our very cute Hawaiian bobtail squid, Euprymna scolopes, and then we have one symbiont, the bioluminescent bacterium Vibrio fissurae. So in this symbiosis, the host is using the light produced by the bacteria for contour illumination to avoid predation. And in turn, the host feeds the bacteria. In this system, we usually work, yes, with juvenile the majority of the time, which are, as you can see here, they are very tiny in scale. And the juvenile squid hatches aposymbiotic, and they will recruit the bacteria from the environment each generation. And they recruit it here in this specialized organ that is situated here underneath the mantle of the squid. And it's called the light organ. You can see here a schematic view of the light organ, where you see that it has two sides, that they are kind of a mirror image of each other, with 3 crypts in each of the sides, which is where actually the bacteria reside within the light organ. So here on the right, you can see a magnification, a TM picture, where you see that the bacteria reside extracellularly along the apical surfaces of the cryptoepithelium of the light organ. And just to remind you all, any time that you have an association with microbes, you always have to have a certain level of communication, not only among themselves, but also between them. For example, during the initiation of a symbiosis, symbiont has to be able to communicate to their host, say, I'm here and I'm a friend. And also the host has to be able to prepare to welcome the symbiont. For example, to orchestrate all, in many instances, developmental changes that happen with the symbiosis, but also, for example, turning down the immune responses towards that symbiont. Because associations with microbes are so evolutionarily conserved, usually the language that is used for this communication is also conserved. And it's mainly due to the recognitions of microbe-associated molecular patterns, or MAMs, that are recognized by host pattern recognition receptors, or PRR. And now we know that in order to this communication to happen, they don't necessarily need to be in close contact between the symbiont cells and the host cell. There, of course, there can be diffusion, but other way that we know that bacteria can deliver bacteria products to their host is through OMVs, or outer membrane vesicles, which are these tiny vesicles that are constantly being produced from the membrane of the bacteria cell. And particularly what we know from the system is that if we just deliver these OMVs, they can trigger normal symbiotic responses like host development in the light organ. So we know in the initiation of the symbiosis, the light organ undergoes huge changes to being able to harbor the symbiont. And it's the process of maturation of the symbiosis. And these changes happen in very different levels: physiological, genetic, and morphological. And from the many years of work in the system from Margaret and Ned, we know even the classical MAMs, or bacteria products, that will lead to specific host responses. For example, peptidoglycan or LPS. Also, particularly of our system, is the perception of the light produced by the bacteria that also induce different host responses. But what we were very interested in is the possibility that small RNAs from the symbiont could be sensed by the host and then induce different host responses. So in that sense, our main question here was, do small RNAs have a role in the interkingdom communication between the symbiotic bacteria and their animal host? And before I get into this, I just want to give a brief reminder of, here you have a central dogma. So if you remember from genomic DNA, it's transcribed to mRNA that is translated into protein. Nowadays, now we know that there are many kinds of RNAs that are also translated from the genome, but they are not transcribed from the genome, but they are not translated into protein. And they are in many, across organisms, very different types that have each a specific regulatory function. And bacteria are not an exception. They also have this kind of RNA that are called non-coding RNAs because they are not translated into protein. In bacteria, they are called small RNAs, and they vary. They have a wide range of size, between 50 to 500 nucleotides. And they can act both at the mRNA level, so for example, binding to mRNA to either stabilize the transcript or down-regulate the expression of that specific gene, but they can also interact with proteins. And these small RNAs in bacteria are very important to regulate bacteria functions, like metabolism, virulence, and environmental and stress responses. So these small RNAs are very important for the normal physiology of the bacteria cell. But how everything started for us, and how we what led us to this crazy path of small RNAs in the system. So everything began because we were doing a pilot experiment for a completely different project, where we were characterizing the circulating RNA from the hemolymph. The hemolymph is the blood of the squid. So in this sequencing experiment, when after sequencing the total RNA population that we were found in circulation of the host, we found a large amount of RNA that was coming from the symbiont in circulation. And within the symbiont RNA that we found there, because we were so interested in the capacity, the regulatory capacity of small RNAs, when we were focusing on that specific group that was found in circulation, it was mainly one small RNA that was becoming the most abundant within this group, and it was called Ssra. And that's how everything started to be focused on Ssra. And before I get into the story of the possible roles that it's having in the symbiosis, I just want to briefly say what it's doing within the symbiont cell. So Ssra is a tmRNA, which means that acts as a tRNA and mRNA at the same time. It's part of the ribosome rescue system of the cell, which means that, for example, when a ribosome is stalled because there is a truncated mRNA, so the protein synthesis cannot continue, it acts as a tRNA, so to be able to continue the protein synthesis, and then uses its own open reading frame to finally finish the protein synthesis, marking this with a tag that marks this peptide for degradation, this potentially truncated peptide. So in that way, now the cell has a free ribosome, which is a very expensive molecule and can keep going the protein synthesis. So Ssra has an important function in the symbiont cell. It's a housekeeping function. And remember that we were seeing in circulation in high levels. So our next question was to go to the source and see where within the light organ do we see this Ssra. And here you can see a light organ section, confocal section of the light organ crypt, where using a highly sensitive in situ hybridization that allows us to directly visualize transcripts. So what you can see here in green is the fissurized cells, the bacteria cells here. In blue, you see the nuclei, the host nuclei, and in pink, you see Ssra. And now, remember that Ssra is a bacteria RNA. So we see it within the bacteria cells. But it was very intriguing for us is that we were seeing it with so much abundance within the crypt epithelial cells, particularly in the cytoplasm of the crypt epithelial cells. So that leads us to question, could be that the host is sensing this somehow? It has any kind of function in the symbiosis, having this RNA so much abundant within the cytoplasm of the cell. So in that sense, what will happen to the symbiosis if then the symbiont is not producing this Ssra, which means that all the signal that you see here will not be there. So in order to address this question, we created a clean deletion mutant of this Ssra, and then we compare different phenotypes, both from the point of view of the bacteria and from the point of view of the host. And the first thing that we needed to know was if this Ssra mutant had any problems of growth. And at least in the media that we look at, rich or minimum media, it doesn't have any problems of growth compared to the wild type strain. And this is due because even though Ssra has a housekeeping function in the cell, Ssra has an alternative way to rescue their ribosome. So that's why we think there is no effect on growth for this mutant strain. Then the next thing that we wanted to see really, in order to see if we could do any of the squid experiments, was is Ssra able to colonize the squid? So here you can see the bacterial levels of per light organ of animals that were colonized by the wild type strain, by the Ssra mutant, or by the genetic complementation, which is just the Ssra mutant that is expressing Ssra on a plasmid. So this is our control there. And what we saw, we were very happy about these results because it meant that the Ssra mutant was able to initiate the symbiosis normally. So we could do squid experiments with these mutants. And so as you can see here, at 24 hours, there is no difference in the bacterial level of this mutant strain. However, when we take it further along at 48 hours, we start to see that there is a problem of the symbiont to persist within the light organ. Also, apart from this problem of persistence within the light organ, we measure the luminescence of the symbionts within the light organ, and we saw that the Ssra mutant strain has a decreased luminance, so it's less bright than the wild-type strain, but only when it's host-associated. So that's another phenotype that we have seen with the Ssra mutant from the bacteria side. But then the most important for us was, okay, so we can colonize the squid with this mutant strain. Does the host care that now is being colonized by a symbiont that is lacking this Ssra? So for that, we did an RNA-seq where we compare the expression profile of the light organ of animals that were colonized by the wild-type strain, the Ssra mutant, or the genetic complementation. And we did everything at 24 hours because it was when we knew we had the same level of bacteria in all our conditions. And what we found is that, yes, indeed, the host transcriptional response changes when the symbiont is lacking this small RNA, Ssra. So here we have a heat map of the genes that were differentially expressed. And what I really want you to focus on here is in this set of genes that are upregulated in light organs that were colonized by the Ssra mutant that happen to be a set of genes that are associated with antimicrobial and immune responses. And actually, we follow up one of them, lactase, because it's an enzyme that is involved in the melanization process of which is an immune response, a common immune response in invertebrates. And here you see again the highly sensitive fish, or HCR, hybridization chain reaction, we call, where we can see a confocal picture of the whole light organ, where in red you see here the expression of lactase, this enzyme that is involved in an immune response of the squid. And here you see that it is an aposymbiotic. And the expression of lactase is just on the crypt epithelial cells. So if this animal were to be colonized, here is where the bacteria could have been. So now if we compare this expression of lactase that we see that is highly expressed in the crypt epithelial cells, when the animal is colonized, you can see here now the symbiont is in green. We see that the expression of lactase is significantly downregulated. Now, if we colonize the light organ with a mutant strain that is lacking Ssra, then the expression of lactase goes back to the aposymbiotic levels, goes back up. But if we add Ssra expression to this mutant on a plasmid with our genetic complementation and we colonize the squid, we see the lactase expression downregulated again to the levels of the wild type. So this means that Ssra is required to downregulate the expression of lactase in the light organ. So this is important because it means that Ssra is required to downregulate an immune response of the light organ that is necessary, it seems, for colonization. So other phenotypes that we look at in the squid is a classical phenotype in the squid vibrio system, which is what we call the cell swelling, which is just the increase in the cytoplasm volume of this set of cells that are just in direct contact with the bacteria, the crypt epithelial cells. And this is a normal symbiosis triggered developmental response. So we, when a light organ is colonized by wild-type strain, we expect to see these cells swelling. However, these cells swelling usually start at 48 hours. And that's what we actually, we can see here. So here we are comparing the cytoplasm area of our animals that were either remained uncolonized, so apo, colonized by the wild type or by the Ssra mutant. And we see that both the wild type and the Ssra mutant induce this cell swelling phenotype. However, when we look earlier on at 24 hours, when we are not supposed to have this cell swelling, we see that the animals that are colonized by the Ssra mutant already have cell swelling comparable to the one of 48 hours. So colonization by the Ssra mutant induces this early cell swelling of the cryptid organ epithelial cells. Now, we don't really know what it means for the light organ to have this early response in cell swelling. But for us, what it suggests is that there is a dysregulation. So if you put this together with what we've seen before, with the increase in expression of genes that are associated with antimicrobial and immune response, then maybe it's suggesting that now that the symbiont is like Ssra, maybe the host is treating this symbiont as a pathogen. So that was actually our next follow-up question, is to ask whether there was any detrimental effect to the host being colonized by this Ssra mutant strain, so the symbiont that is lacking this Ssra. However, in order to keep going with this question, we really wanted to distinguish between are all these phenotypes that we are seeing due to the lack of Ssra? So if Ssra is a potential signal, is due to not having this signal within the host cell? Or is just due because now the host is being colonized by a symbiont that doesn't have Ssra activity, which again is, even though Ssra has an alternative way to rescue the ribosome, we don't really can emulate all the situations and the conditions that the symbionts see within the light organ. So maybe not having Ssra activity was important for the symbiont within the light organ. So in order to keep going with this project, we really needed to tackle this question. So for that, we, if you remember, Ssra, in order to have its activity within the bacteria cell, requires a chaperone called SMPB. So what we did was to create a clean deletion mutant of this chaperone, that this mutant will produce Ssra normally, but does not have Ssra activity within its own cells because it's lacking the chaperone. So with that, we also checked that with this mutant, yes, indeed, here you can see again another section of the light organ where in green you see the SMPB mutant, and in pink you see Ssra. So we see that this mutant strain is producing Ssra, and it's also getting within the symbionts, within the host cells, sorry. But we know that this mutant strain will not have Ssra activity. So knowing that, and in order to ask whether it has any effect on host survival, we did a survival assay over the first few days of life of the squid that were colonized by either the wild type, the Ssra mutant, the genetic complementation, or the SMPB strain. And now, just a reminder, this strain will have Ssra, but doesn't have Ssra activity. So what we saw with the survival assay is that here in red, you can see the animals that were colonized by the Ssra mutant that die earlier than any of the other conditions. And because we are not seeing any difference between the SMPB mutant and the wild type, it means that this phenotype that we are seeing here, that is kind of weaken of the host, that is dying earlier than the others, is due to really the absence of Ssra potential signaling, but not to the lack of Ssra activity within the symbiont cells. But now, again, another point, why we are seeing this dying off, this early dying off. So it's not that now suddenly the Ssra mutant has become a pathogen and has acquired out of nowhere a pathogenic island, right? So we really wanted to know why these squid are dying earlier when they are colonized by this Ssra mutant strain. So because we are seeing that there is an increase, what we think is an increased immune response with the dysregulation of having also a earlier cell swelling phenotype. That what we think will have an energetic cost. And it happens that the juvenile squid live out of their an organ here that is called the internal yolk sac, which is a reservoir of nutrients that they use up over the first few days of life until they learn to hunt and they are able to feed for themselves. So because this internal yolk sac is so rich in lipids, it's very easily imageable by confocal microscopy, just with a lipid dye. So we did that. We decided to measure the difference in the size of this organ, whether it was right after hatch, which is the biggest that this organ is going to be, or after 2 days after hatching of animals that were either colonized by the wild type or by the Ssra mutant. So what we saw here is that after 2 days of colonization, yes, we know that we see that the animals are indeed using the internal yolk sac when the animals that are colonized by the wild type strain. However, when they are colonized by the Ssra mutant, we see that they use the internal yolk sac more rapidly. So the absence of Ssra causes a rapid depletion of the internal yolk sac. Maybe that's why they are the host is less robust and they are weakened and they die earlier. And so we've seen many phenotypes that are all focused on what happens to the host when it's not sensing Ssra. But in normal situation, when you have your wild type strain or any strain from the environment, Ssra is there. So how does the host detecting this Ssra? So first of all, and I didn't have time to get into this, so I'm sorry for that, but our hypothesis here of how Ssra is being delivered within the crypt cells is through OMVs, so through these outer membrane vesicles that are produced from the membrane of the cell. Because here there are several things. One, we know that these OMVs are uptaken by host cells and they stay in the cytoplasm of the cell, and they can induce normal symbiotic development in the light organ by themselves, so only delivering these OMVs. Also, we saw that so it really doesn't matter in which media you grow the bacteria, or in which phase of growth the bacteria is when you purify the OMVs. We like in all the media that we tested, or growth phase and everything, we've always seen Ssra that is carried within the OMVs. And also another thing to mention before I move on is that we check the RNA content of the OMVs that were purified from the wild type strain or from the Ssra mutant strain. And we didn't see any differences in the RNA content of these OMVs, except the presence or absence of Ssra itself. So with that, in order to answer the question how is detected within the host cell, we used as a proxy hemocytes. Hemocytes are the immune cells of the squid. And we use hemocytes because they uptake OMVs in large quantity. They are very easily imageable and very easy to work with. So with this model, what we did is we culture the hemocytes from the squid, and we then deliver either wild type or Ssra OMVs. And now remember that we know there is no difference in the RNA content of both OMVs except the presence or absence of Ssra. And also we check at least the major, as far as we know, the major proteins or the proteins in large amounts within these OMVs are also the same. There is no difference. So that means that we can deliver these different OMVs to the hemocytes and then say that their response is really due to the presence of absence of Ssra. So we did that, and by QRT, we measure the expression, the change in expression of several candidates. And we really wanted to focus on, for example, genes that are related to antiviral response, right? Because we are talking about an RNA. And particularly, we were very interested in a cytosolic RNA sensor called RIC1. And actually, when we deliver wild-type OMB, so the ones that do contain Ssra, to hemocytes, these are the only ones that have shown an unregulation of the cytosolic RNA sensor, which suggests that Ssra might be detected through this host cytosolic RNA sensor. So putting this all together in our model, we know that when the light organism is colonized by the wild-type strain, so a strain that is producing light normally and is also delivering its OMBs that contain Ssra, most likely through interaction with RIC1, we see that then there is a normal response in the symbiosis. So you have normal immune responses to keep the symbiont in check. So it seems like there is an immune response modulation to the symbiont being colonized, to the colonizing symbiont. However, when the light organ is colonized by a mutant strain that is lacking this Ssra, then we see that because you don't have Ssra within the cytoplasm and there is no immune modulation, we see that the symbiont has problems to persist in the light organ. It also has decreased luminescence, and the hosts we've seen increasing expression of genes associated with antimicrobial immune responses. We've seen also premature cell swelling, a rapid depletion of the yolk sac that overall leads to a decrease of host robustness. So this is a clear example of how a very, very tiny RNA molecule can have huge consequences for both the host and the symbiont in this system. And for future perspective, so we know there are many things that are still a work in process in the system. We know a potential mechanism or detection method with hemocytes, but we really don't know which are the underlying mechanisms of this Ssra sensing within the epithelium of the light organ crypt. Also, we don't know yet how is the downstream signaling of this Ssra after it's been detected. How after detecting Ssra you see this immune modulation that you don't see if you don't have this Ssra. And another possibility that we are also very interested in is the possibility of, can the host detect Ssra from different bacteria? So the same way that we can use, for example, 16S as a marker to see which species of bacteria do we have, could be that the hosts are able to distinguish Ssra from specific bacteria. So in that sense, for example, if we were to colonize a squid with the mutant strain that instead of being expressing the same Ssra anaplasmid is expressing, I don't know, E. coli or Pseudomonas, any other smaller Ssra from other bacteria, we'll be able to complement them and then get at the level of the white type as we are seeing with the Ssra1. Or it will consider that this is not the symbiont that is supposed to be there. And lastly, we are also, now we kind of opened a new window to this world, to this RNA world. So do we know if we have any other symbiont RNA that are getting into host cell and can potentially also lead to maybe different response in the host? or could they be monitoring different states or metabolic states of the bacteria, for example? So with that, I just want to leave you with this kind of teaser image, confocal, where you again see a section of the light organ crypt, where now in green you see the Ssra, so you see that is highly abundant within the cytoplasm of the cell. But this time in pink, you see other small RNA from the symbiont. So it's another bacteria, small RNA, that we see has a completely different pattern of distribution that Ssra. So we always see it in this localized within the nucleus of the epithelial cells in this punctuated way. And well, we don't know what it really means for the host yet, or it has any similar different host responses as we have seen with the Ssra. But it has certainly opened a new point of view in the system with the possibility, all the possibilities that we have with RNA. And so there is much, really much that we don't know, but hopefully I managed to convince you that small RNAs can be an additional way of communication in host-microbe interactions. And with that, I would like to thank Margaret McFall-Ngai and Edward Ruby, and also all members of Margaret and Ned Labs, current and former, which there are plenty, and it's very difficult to fill them all. But particularly, I would like to thank Clotilde, Tara, and Eric that really helped push forward this research. And I also would like to thank Frederick Packett, that we were extremely lucky that he decided to do a sabbatical in Hawaii with us. And we really enjoyed all the discussions that we had with him. And also I would like to thank my committee members and of course all of you for your attention.